Welcome to distributed cancer cell typing and tumor purity. I know it's the end of the conference. I know everybody's had a full day, but I hope we're all excited about computational histopathology. Who's excited? Anybody? All right. You are excited. That's good. Thank you. All right. I I'm Robert Kramer. I'm a principal data scientist at Providence Health. I've had a long, winding path, and I'm thankful to be here. If you don't know, Providence Health is very, very big. We have over 29 million patient visits. And I know in most cases, health organizations and researchers don't have enough data. We have entirely too much data. So we have the opposite problem. And then, the thing that we like to point out always is that this 2.1 billion in community benefit. So Providence Health we're a Catholic nonprofit healthcare system, and we're here to help the poor and vulnerable. So that 2.1 billion is, things that we give to help those people. All right. Got a little stragglers. We're good. So let's talk about histology. Histology is the study of microscopic structures, and it's typically been historically an analog field. So, we have like actual microscopes, actual tissue, actual slides, and that really persists to this day. So most leaders in like a pathology lab would think that storing a digital image would be more costly than storing the little slide in a warehouse. And then they also have a duty to keep care of this thing for like 10 years. And for us, we're trying to get into digital pathology and digital histology. And we're looking at how do we get those workflows to happen. And when we look at cell typing, it's really like one of our first steps towards understanding the tumor microenvironment. And, understanding the tumor microenvironment is how we understand how to get those immune cells or those lymphocytes to attack our tumors and, hopefully like cure cancer. So we measured tumor purity in this case, and really that's because we wanted something that would help with our TrueSight oncology genetic sequencing workflow. And so tumor purity is just the number of tumor cells or the number of total cells. It helps with consistent objectivity. It helps with quality control. All right. The one big takeaway from this is getting anything going in our production environment is extraordinarily, there's a lot of things that we didn't expect. There's things that are difficult, and this is like a big challenge for us, and it's also a big achievement. And this will help us use much more complex in our foundational models in the near future. It's the same basic process. We also learn how to use these tools. So, we need to be able to do annotations in the cloud. We need cell viewers for our pathologist. We need to have model monitoring. We need feedback in an annotation engine. So these are all things that, we can learn with something like cell typing in a genomics workflow in order to understand all the problems before we get to something that might be like an actual clinical model. Okay, I'd also like to talk for a second. Prov GigaPath, it just came out 2 or 3 weeks ago in Nature. It is the product of like 5 years of partnership with Microsoft and University of Washington. It's the world-leading computational pathology model. It's leading in almost every metric. And this deployment is fundamentally the same as the cell typing deployment we'll talk about. So we have some initial things I've already kind of got in there, and I'm working very hard to kind of get more. There's open weights. It's on Nature, GitHub, Hugging Face. You guys should all check it out. It's pretty awesome. Okay, let's talk about like this case study. So we looked for a model that we could use and that we could figure out how do we actually get to production. And this is a relatively straightforward supervised learning. It's split into 3 classes. We have stroma, lymphocytes, and tumor. And our lymphocytes, those are the immune cells that attack our tumors. Stroma is really, in this case, it's kind of everything else that's not tumor. But, tumor is really important to understand, to know, like, do we have enough material for our TrueSight oncology Illumina, genetic variant pipeline? We had our pathologists do non-exhaustive annotations. Getting pathologists to do annotations, we're very grateful that we had some, but it's not an easy thing to do. And they couldn't really go and annotate each cell. So what we have is we have these regions, And there's some issues with that, but it's been good enough in this model. And in general, what happens is you'll split that whole big whole slide image, which is a gigapixel image. There's like 30,000 regular sized images in that image. And then we'll split those into tiles and we'll send them through a pipeline that finds the cell centroids. We use StarDist for that. That's a whole deep learning program that's just in our pre-processing. And then after that, we take our cell centroids, we center our tiles on the cell, and then we use the whole tissue to predict it. We know that sometimes there's other cells, other types of cells within it. We get okay performance, good enough for the type of tumor purity calculations we're trying to get. And in the end, we get these little colorful things. You can see all these cells. It's fun. Okay, tumor and total cells are predictive. Here we can see that indeed our total cells are highly correlated with our DNA stock quantity. So this is really good for our quality control. If we have too little DNA, if we have too little stock, then we're not going to get a reliable run. And then we can also see the average clinically significant allele frequency is increasing with the amount of tumor. And this is something that we would expect. We'd also expect to not be such a high correlation. There is a little issue over there with the actual DNA stock quantity. In the real workflow, it's not digital, it's analog. So they're literally circling a slide, and that's where they take the tumor from. That's where they take the material for the genetic sequencing. We don't quite have that. So in the actual modeling, we have to sort of model the difference between the amount of tumor in the circled region versus the amount of tumor in the whole slide. Okay, so we have pretty big challenges here. We have hundreds of thousands of whole slide images that were scanned as part of that Microsoft research project. That's why Prob GigaPath is such a good model. But for us, just doing the cell typing, it means that if we wanted to do historical analysis, we need something that will scale to hundreds of thousands. As I said before, there's like 30,000 tiles in one image, and that's on average. It can be more or less. And what's really important here is that we have to finish this in at least enough time to do 20 new cases per day. And naively, when we ran just the research model on Databricks, well, at first it wouldn't even run, but you get it to run, and it took like 3 hours. We'll find out. Most of that is I/O. We could probably solve that now, and it's going to be less just even with that, but it still wouldn't scale to the type of scale that we need. All right, so let's talk about Databricks and like what this whole story was. Databricks has been an excellent partner for us. They provide excellent representatives. They put us in touch with SMEs. They get us access to these accelerators. But as I talked before, we discovered this model. We were looking for a computer vision model. We were looking for something that we could get into production. And that was sort of like my job, going to work with the molecular genomics lab. And once we found one, Databricks was a great partner. Then we built it, or I built it. But we go through and we adapted it to PySpark. And because we already have our TrueSight Oncology running in Databricks, it's trivially easy to join by a session number or join by path case ID. It's not a one-to-one join, but it's close. All right, so if you are going to take a research model and put it into production, this is the kind of thing that you're going to have to do. So your researchers, they're like me, they're like everyone. Like, we learn how to do things the regular way, which would be like in PyTorch with one GPU and a local hard drive and a single machine. Maybe you're on the cloud, but you're not really doing cloud stuff. And from there, we're going to blob storage. That blob storage, we're orchestrating with the Delta Lake. So you can't put a whole slide image in a Delta Lake, but you can put a path to a whole slide image to a Delta Lake. And you can put the binaries of the tiles into a Delta Lake. And then we're going to run it in Databricks. We got PySpark, and we send them out to our little executors to do all our independent operations in parallel, and we can track it all with MLflow. Okay, and then here's kind of what the planned production workflow looks like. I mean, right now we have this Hamamatsu scanner scanning new images, and that is picked up by this digital slide archive, which is a cloud viewer. And then you can run this, and you can join it to the TSO 500 results. And then we have an internal app that people who monitor the workflow can look at it. We need to do studies before we actually get this into it to where it's the pathologist sees it before they go. But this is, in general, all the engineering is done here. All right, and then the source code, because we can use repos and arbitrary files in Databricks now, it kind of looks like a regular repo and it looks like regular software engineering. So I really appreciate that. But in general, we have our classes and it helps us with the modular code, and this will help us, distribute and do new things in the future. Okay, now let's talk a little bit more detail about like a typical inference run. What we start out with is a preprocessor object, and that preprocessor object will create a meta WSI DF. So this meta WSI DF, it makes it all the way through, and it keeps track of what we're going to train, where the annotations are, what we're doing inference on. It says where the paths are. There's stuff in here like you got to have like a Spark path, and then you have to have like a regular Python files path because dealing with local files in Databricks is a little tricky. But that gets passed to all these other objects. And so that's really what's central to the workflow. Then the next thing we do is we prepare this max extract. This is simple thresholding, but we don't want to do tiling on everything. We want to, like things like the white space, we want to make sure that we're not including that. So we're trying to just get the tissues. And then the centroid thing, the centroid DF, what this is doing is it's kind of broadcasting and running StarDist on all of our executors. And it's doing that on, we'll talk about it, but on the cache WSI. And so that gets us the cell centroids. And from there, then we generate a patch for each location where the cell centroid is. And you can save that as a binary in Parquet. So it's not too big. If you try to materialize it, you'll, as like, let's say a NumPy vector or something, sometimes it's a little too big. But in general, like it's pretty good. And then if we talk about like how we actually distribute it, it's a pretty brute force apply. And this repartition, what this allows us to do is kind of say how many times am I going to split this up? How many different pandas data frames am I going to have that we're going to operate on? And I found that if you make this higher, like a multiple of the number of CPUs you have, you do a better job of handling, like, let's say if you have a really big WSI or if you have a really little one. So it's it helps with that a little bit. And basically, we pass this through, it sends it out to all the executors, and then they all happen in parallel. Okay, here's a visual representation of the same thing I was just talking about, and hopefully it gives you an idea. Sorry, I should have had my microphone a little closer. I apologize. This is the first time I've been at a conference, so thanks, everybody. All right, so we have all the little executors here, and these are all theoretically independent operations. So you can do those all at the same time if you'd like to. There's some reasons why you don't necessarily want to do that. And then we also have to keep track of which tile it is because there's some overlap issues, and we'll talk about that in a second. But at the end, you get this pretty picture. You can say, hey, these are lymphocytes, that's a tumor, these are stroma. And yeah, it's useful. Okay, let's talk about our I/O issues. Let me make sure I didn't screw it up. Okay, so the thing that happens if you're using OpenSlide is kind of weird, but it's operating in this pyramidal TIFF sort of format, and it wants to know like, hey, where are all the things that I need to open? So it doesn't actually pass data, but it opens up a ton of objects, like let's say 10,000. And if you're on a local like hard drive, this doesn't cause a problem, but those concurrent like opening things, those really, really clog up blob storage. So it's very, very difficult to actually use OpenSlide on here, and you get like terrible, terrible performance. And so what we did is I looked through, I don't know if you guys have heard of Petastorm, but it's like a deep learning thing, and it does some caching. And in there, they use the disk cache. And so that's kind of how we figured that part out. And disk cache will handle these concurrent operations after we copy it over to the local executor. And it'll also delete new ones when you don't need the old ones. But, it's relatively simple, but including this in there, like this increases it like maybe 10 to 100 times faster. And that it's really a lot about this when you're talking about the deep learning stuff on Databricks or Spark. All right, another cool thing we do, I don't know if any of you guys have done geospatial stuff, but we actually get our annotations as GeoJSONs, and I did geospatial, I guess, when I started at Providence. And so there's a new Shapely 2.0 that is vectorized. And so instead of saying each individual cell, like, where are you? Are you in there? Are you out there? You can do the whole region and just all the cells at once. Are you in? And this still can distribute, and it distributes across with that pandas operation. And then, it's all good. Okay, and then the other kind of thing that we had to figure out was how to distribute the StarDist. So StarDist has a real nice predict big function, and it'll go across and it'll do a whole WSI for you. And it does that with kind of like the overlapping tiles as it runs across. And I couldn't quite implement that. It didn't exactly work out for me, but I found it much easier to create extra padding, at least enough padding to be more than one cell, and then use these unique tile IDs in order to make sure that we only have one prediction within the region, because you get all these overlap things. So we had to deal with that. But what that means is that we can do these small predictions all in parallel. And, like theoretically, you could do, if you had that many CPUs, as many tiles as you have. So you had 30,000 CPUs, you could do the whole thing just like immediately. Okay. Another thing is just like for these experiments or ablations, it was nice to be able to use MLflow for it. And it's nice to have a smaller sort of data set. It's nice to be able to go and like define your pre-processing. We also use like Pydantic just to say like, hey, are you using one of the experiments we talked about? But it's pretty fun. And, the results are pretty good. For the same kind of compute cost, one we ran with the GPU and one without, the StarDist cell detection is faster. And then that cell typing inference, this is a worst case scenario, that 164 minutes. That's without any of the disk cache. That's not just because the GPU doesn't really get heavily utilized because of the IO issues. But we do get it down to 4 minutes. So, it's about 9 minutes per slide on a reasonably priced compute. And then it scales roughly linear. So this 30 executors, we got down to 1.8. You can obviously get much larger than that. There's some overhead because we're copying over those WSI files to the local cache, but that's what blob storage is very, very good at, is copying like a 2 gigabyte file. So that only takes a few seconds. And just to recap, that disk cache really helped us, and we'll integrate very easily with our genomics platform. And I've already started to use the same basic ideas in order to get our new like fancy foundation model kind of running. So anyhow, I hope everyone had a wonderful time, and I'd like to thank the Early Child's Institute. This is sort of where the researchers were. The Molecular Genomics Lab have been excellent partners. I'm in healthcare intelligence. My boss is Lindsay Myko. Thank you for letting me work on this stuff. And if there's any questions, we have a couple of minutes.